I saw from the program I'm supposed to talk about the synthetic side of vision. So I thought this would be a good opportunity for me to tell you my personal thoughts about how I believe space is encoded in the brain. And the implications for that are how we need to replicate the mechanism of encoding to build machines that perceive space in the same ways that humans do, or what we should take into account when we build prosthetic devices in order to make them work with this kind of machinery. Okay, so this is my personal view. I remind you that with Martina, I direct this laboratory, the Active Perception Laboratory, and we both pour resources into this group of students. We are funded by several sources, in particular NIH, and contact with the industry, Meta. Okay, so the essence of the talk is that if we really want to understand how space, and again, this is my personal view, how space is represented in the human brain, we must study it together with motor behavior. There is no other way around. And the reason for that is that they are intimately, tightly bounded. You can't take them apart. Now, we all know that as we explore the world, we are moving all the time. I'm moving here, I'm moving my eyes, I'm moving my body. I live in a place, usually my office, in which nothing moves, but everything is moving on the retina because I move all the time. If we were talking about other sensory modalities, the fact that you need to take behavior into account when you study sensory perception is obvious. Think about olfaction. If I want to smell an odorant, I need to sniff. And I need to take, my brain needs to take that motor action into account when it interprets the signals. If I need to make sense of my somatosensory signals, touch, right? I need to take into account how my hands are moving, for example, in order to then reconstruct what is out there. So the signals, the sensory signals, needs to be interpreted in the context of motor information. Not only that, but also motor information is generating those signals. The way these signals are shaped depends on the way I move my fingers on objects, right? Same thing for the sniffing action. But in vision, we tend not to think in this way. And that's because we have this nice machinery that creates an image. It makes space, it makes spatial representation explicit on the retina. And so we think that that's sufficient for representing spatial information. The standard idea is that everything works as an image, as a camera in a way. And the way we represent space is because different receptors and then different neurons are located in different regions of those images, have receptive fields in different regions. So the topology of these maps is where space is represented. I ask for a favor, is there any way we can move Martina down or somewhere in the corner? Because I'm going to acknowledge the students who did the work. You know, I talk about this, but the work is done all by students and postdocs, and they will appear from time to time up there. So I don't want them to get obscured. All right. So let's think about this point that I'm trying to make a little bit further, right? I think in vision we have focused too much on images and without taking into account behavior and without taking into account another dimension that behavior brings in, which is time. For example, how is a scene represented in the brain? The standard view is that we do more and more sophisticated operations as we go up in the hierarchy. Initially, we start simple features, then maybe in the visual cortex, we start extracting edges, and we get a nice image like this, and so on and so on and so on. Or think about stereopsis, right? How do we encode information in depth with the two eyes? Well, everything is based on precise correspondences. An object is falling in this position on the retina and this other position on the retina. The two images are compared, and by triangulation, we get depth. Or even if you think about diseases, like, for example, a very common one, myopia, which is endemic at this point. Why is the eye not stopping growing at the right point? Well, all theories out there are based on the images on the retina. Well, it's blurred. And what is the sign of blur? Well, you're going to have different types of blur, different types of anisotropies that will tell you whether the eye should go in one direction or the other. Everything is based on images. But we know that this picture, from which we have learned a lot, is incomplete. Because we know that those neurons out there that are doing something interesting responds to time, not just to space. And we know that our eyes are always moving. In fact, what are even corresponding points on the retina where the two eyes are not moving exactly in the same way? So that becomes an interesting question. So our eyes are moving, and those neurons, here at the mouse, are also responding not just to spatial, they're not selective not just in space, but they're also selective in time, the way you present that stimulation matters. In fact, we know that the temporal system itself, the visual system itself, is very sensitive to temporal changes, right? These are old measurements from the 60s of temporal sensitivity. You know, we have a range of things that change in time that we respond best to. I'm assuming everybody can read these graphs, right? What is temporal frequency? Okay. Because I will be showing graphs with joint spatial and temporal frequencies, but I'll get to that later. This happens also in macaques. They tend to prefer things that move. It doesn't matter how you stimulate the visual system. Even if you look in the fovea, which is considered something that responds more to stationary stimuli, that's not true. It's still responding to temporal changes. Here is an example of a study in which you are modulating a stimulus fixed on the retina, right, as the retina is moving, and you're measuring what is the temporal sensitivity of that patch of the retina, different lines at different regions within the foveola. And you see everywhere in the foveola, they respond best around 5 hertz or so, when you modulate 5 hertz. Some people say, well, yes, it's still responding to static stimuli, but is it really? I mean, I don't really have a way to present a static stimulus. At some point, I need to turn on the stimulus. So how do you measure sensitivity to stationary stimuli becomes really important. But I would argue, because I'm giving my own perspective, that there is no vision at 0 hertz. And this comes from the classical literature. These are old experiments in which people stabilized images on the retina, the so-called suction cup. These are sort of big contact lens that you put on the eye with a pump. Then you make vacuum, so it sticks to the eye. And there is a pole with a mirror. So you can project an image on this mirror, and then when the eye moves, the pole moves, and it stays stationary on the retina, right? And when you do that, the image will fade away. Eventually, there is no vision. You can see yourself that you're not sensitive to very slowly changing things, because this is not an image, it's a movie. It's changing continuously, but it's changing so slow that you don't realize. You probably saw that it resets itself now. And so it's just gradually changing, and when it goes very slowly, you don't see it. We don't have sensitivity to 0 hertz. We only respond, I will make my case, to things that move in time, they change. My second point is that most changes come from us. All changes come from us. In my case, where I spend my days in an office where nothing moves. And in particular, they come from eye movements, because I'm making different types of eye movements, particular saccades, every few 100 milliseconds, as we all know. Saccades, by the way, are the most frequent type of behavior that we perform. They occur more frequently than a heartbeat. In the time we spent together here, you would have made 20, 30,000 saccades. Imagine that. In the lifetime, in the span of a lifetime, you make billions of saccades. So we make saccades all the time, every few 100 milliseconds. And even in the periods in between saccades, we keep moving our eyes, as we saw with Martina. And we have small saccades. I believe this is just a misnomer. It just comes out from historical reasons. We can talk about that later, but they're just small saccades. And then in the middle of saccades and small saccades, microsaccades, you have this jitter, this slow motion, jittery motion of the image that I will call, again, for a lack of a better term, I think that's a misnomer too, ocular drift. Okay? So whenever I refer to ocular drift, I'm referring to this slow, meandering, jitterish mode of motion that you see inside. They're very small movements. We're talking about 10, 20 minutes of arc. Your fingernail at arm length is 1 degree. So 10 minutes of arc is 1/6 of your fingernail at arm length. And here it is. Okay, good. So normally, in the standard picture of how space is represented in the brain, all this motion creates a problem. How come the image doesn't get blurred? How do you avoid it? How do you suppress it during saccades? And so on and so on. But I would like to make the argument that we can revert the point and in fact see this as an efficient strategy for representing visual information. And this is my thesis. So here's what I'm proposing. And to do that, I'll go to a two-dimensional world, like in Flatland, right? Let's live in a world that is based on a plane. And we're looking out there. Here is our visual scene as we look out. And I'm looking at one point of this scene. Here is my retina. And as I'm moving my eyes, if I don't move my eyes, the input to the retina will not change. So different receptors on the retina, one here, will keep having in time the same input. I'm not moving my eyes. But if I do move my eyes, then things will change. So if I don't move my eyes, obviously the only way in which I can reconstruct what is out there is by integrating information across receptors, by looking at the image, which is the standard traditional way in which textbooks of vision are based. But if I do move my eyes, what I do, I modulate the input at each individual receptors. Essentially what I'm doing, I'm transforming the spatial scene into spatiotemporal flow. Spatial information is conveyed in the joint space-time domain. Now I have many strategies to extract spatial information from here. In the very limit, each individual photoreceptors will tell me something about how the scene is changing in a neighborhood, okay? So I will argue that we use this, and I call this active space-time encoding. That's the proposals that humans encode space via temporal changes that they themselves produced, and an obvious case is eye movements. So self-motion yields temporal modulations that contain spatial information. Depending on the way I move, part of this information reaches the temporal range of sensitivity of the visual system, and we use these modulations to create spatial representations. A couple of reviews in which we hinted at these things. We probably need to write a more comprehensive review, but anyway. So the starting point for me is really to look at what is this retinal input. So let's start from there. Retinal input in an active observer. And here is where I like to introduce my preferred way to represent spatial information in the temporal domain. I will move, I'm an engineer by training. I love to think in terms of frequency, spatial frequency and temporal frequencies. And so that's what I will do here. So we are all familiar with spatial frequencies. Slow changing gratings, high changing gratings are represented on this axis, right, as different points on this axis. But as we move, we are transforming these patterns into temporal modulations. And so I will have another axis which tells me how these patterns are changing, the temporal frequencies. My behavior will bring them up somewhere, these different patterns, and so that they become temporally modulating gratings, right? Each point, every point in this joint spatiotemporal domain will be a grating at a certain spatial frequency that is modulated at a certain temporal frequency. Higher up will be faster pulsating, right? Lower down will be slower pulsating. Okay, how does behavior transforms this spatial pattern, these images, into a spatiotemporal flow? Well, a good way to start is to think about eye blinks. And that's because eye blinks are a particular easy type of transients. They are easy because they affect all stimuli out there, the entire scene, in the same way. They blank out the image, and then here it appears again. So both low spatial frequency and high spatial frequency gratings are undergoing the same kind of temporal changes. Good. So here they are. Both low spatial frequencies and high spatial frequency will be transformed into temporal changes in exactly the same way. We are modulating the same way. And what I will do to make things easier to represent things, I will take into account the temporal sensitivity of the visual system. We saw before that the visual system is not uniformly sensitive to all these changes, but there is a range of temporal frequencies where the visual system responds best. So what I'm going to do, I'm going to integrate all this information weighted by the temporal sensitivity of the visual system so that I can have an easier graph to represent things. Here it is. I'm going to have just one axis, which is the spatial frequencies, And on this axis, I can just tell you what is the strength of the input signals that I'm producing. If I'm right here in the middle of highest strength, it's going to be very strong. If I'm up there, it's going to be weak. So I'm just going to plot the strength of the temporal modulation as a function of spatial frequencies. You're talking about a blink. It doesn't matter what grating I present out there, the temporal modulation is always going to be the same. So it doesn't change, it's uniform, right? That's why I said eye blinks are a particular case, simple case of transients. Let's think about another type of movement, a movement that makes the scene translate uniformly, a uniform speed across the scene, maybe a smooth pursuit, something like that. Then in this case, if you translate the stimulus, it turns out that it's not going to be uniform anymore. High frequencies will go to very high temporal frequencies, and low frequencies to very low temporal frequencies. So there will be a range of sensitivity that creates a sort of bandpass shape like this one. And not only that, but depending on the speed at which you move, you're going to change the tuning of that curve. I can give you an example of this. If you fixate here on this dot and you keep your eyes on the dot, you may see a grating. But if I start moving the grating and you follow it, you may see that there is another image down here. Hope this is working, depends on the projector, depends on where you are relative to the projector, but you should be able to see a face. So what is going on here? Well, that image is composed by two images. There was a grating, and there was at a low spatial frequency an image. We are moving our eyes, we are pursuing the dot in this way, we're moving our eyes, uniform velocity. So what we are doing, that corresponds to rotating this axis by an amount that depends on the speed of the eye. So this grating at low spatial frequency here becomes available at low temporal frequencies. This other grating here at high spatial frequency becomes available at very high temporal frequencies. If we move the eyes at the right speed, this will go very high. And now what we are doing, we are unmasking, we are bringing it out of the region of sensitivity, so we are unmasking the phase from the grating. We are actually bringing the phase toward the higher region of temporal sensitivity and the other one away. So when I show these kind of demos, people get really surprised. But hints of these demos are there in the literature since a really, really long time. We can go back to the 1980s in which people measured sensitivity to translating images. And they found that, first you have this bandpass shape in sensitivity, which depends on the speed at which objects are translating. If you translate it at different speeds, they all move. But then if you look at the temporal frequencies that they are generating, it's always the same. Meaning, what seems to really matter for what you see is what is reaching the right range of temporal sensitivity. The essence of these ideas that was there in the 80s is what I'm trying to debate today, with the difference that I will argue that eye movements do this for you. Okay, so middle summary. And again, stop me at any time. And also, I don't know if I put too many slides, so stop me when you're tired. We can just stop there. Just take a break. The visual system is primarily sensitive to temporal changes. That's point number one. Most of these changes, almost all of them, originate from the observer's own behavior, particularly movements. Of course, when motion originates in the scene, that's really important, and we have to pay attention to that. But most of it generates by myself. As the stimulus moves on the retina, space becomes transformed into temporal modulations. And spatial vision, I would argue, depends on the information that is delivered in the range of where it is behaving. Critically, the way I move determines what becomes visible, determines what spatial information reaches the range of temporal sensitivity. So let's look at some type of eye movements. And after blinks, perhaps the easiest type of behavior is ocular drift, the jitter that you have in between saccades, right? Okay. We're not aware of doing this, but there are nice demos that make us realize it. You may see this central square moving relative to the other, to the surrounding. That's caused by your own eye movements, your own head movements, the retinal motion that together they are generating. And this guy here is a master in creating these kind of illusions. The standard, all classical textbook view of drift is that, oh my God, you can't even see the image. There is a face. Maybe you can see the face. It's very faint. Okay. But if you fixate carefully, the image will go away. And the original classical idea is that that's because by fixating, maybe you're suppressing your microsaccades. I don't bite for a second, by the way. But that's the idea. And so in textbook, you'll find, well, you need this drift to refresh neural activity. The point that I'm trying to make today is that you need this drift and all the other eye movements to structure neural activity. In fact, to structure the visual input so that it will respond to spatial information. Okay, so let's go to drift. Oops. Yeah, we still have Martina on there.
Yeah, unfortunately, yeah.
Okay, so it's like for what we saw before for a uniform translation. The spatiotemporal reformatting from drift depends on the spatial frequency of the stimulus. Let's work out this together. Let's take, for example, an intermediate spatial frequency, and here is a receptor on my retina that is a recorded trace of eye drift. And we can see the temporal modulations that it creates. Good. So these temporal modulations have a certain amplitude. There is a certain strength in these input modulations. But let's change the stimulus and take this exact same eye movements, but now the stimulus is at lower spatial frequencies. Same eye movements, but lower spatial frequency means that the temporal modulations are going to be much smaller, right? Not much change here. Or if I take a higher spatial frequency, I get as large as a change as in this case, but now they're going to be much faster. So in the range of sensitivity, of temporal sensitivity of the visual system, well, these are too fast. These are maybe good. This is just too little of a change. Okay, so in the range of temporal sensitivity, I will get a sort of bandpass. Here it is. Sensitivity that peaks around a certain frequency. And you can model this very well. This is where it's coming from, by means of Brownian motion. So you can model the eye jitter by means of Brownian motion. And this is what you get will be the power of those temporal modulations. Power of temporal modulations means for me the strength of the input signal. Okay, so there are a few interesting things about this picture. The first one is that the jitter of the eye acts as a sort of spatial filter. I'm filtering the image, right? I don't have low spatial frequency produce little changes, high spatial frequencies produce little changes that the visual system can respond to. There is only an intermediate range that provides a signal that is strong enough for the visual system to respond. I'm filtering the image as I would do with a filter in space, but I'm doing that in the temporal domain. Second, it's a filter that enhances high spatial frequencies. See, there is a suppression at low spatial frequency. It's a very strange filter in a way because it just happens that it's growing in a quite large range of spatial frequencies proportionally to spatial frequency. The power is proportional to spatial frequency squared. You have to remember that we live in a world in which the contrast of the images is structured exactly in the opposite way. There is a lot of power at low spatial frequencies, little power at high spatial frequencies. When I look at this natural world by jittering my eyes, what I'm doing, I'm creating temporal modulations that are all the same across a broad range of spatial frequencies. I'm equalizing them. It's called whitening. And parentheses, whitening is a standard technique in signal processing to improve the efficiency of representations. I won't go too much into this today. Topic for a separate talk. It's interesting that this behavior is sort of creating this equalization, which is a form of matching, you can regard it, between the luminance modulations from ocular drift and the natural visual world. Okay. Another thing that is interesting is that if you change the characteristics of your drift, you can actually shift this filter. If you enlarge it, you can shift this filter toward lower spatial frequencies. If you shrink it, you can shift it toward higher spatial frequencies. So you have a way to decide how you're going to filter your image, which spatial frequency I can enhance by moving your eyes in different ways if you have control of your drift. And I will tell you that you have. Okay. So all this is a nice, if you want, mathematical characterization. Maybe a bit boring at this time of the afternoon, I don't know, but it would be irrelevant if these modulations weren't important. So my goal now is to convince you that they are important. Obviously, you need to have good systems in order to, good eye trackers in order to record movements at this scale. Later on, I will show you movements that you can record more easily and for which you can apply the same principles. An approach that we use in the lab is through this coil-based system, which subjects can move freely. They have coils in their eyes, but now they can do real-world tasks. And they can point at objects in the real world, or they can interact with objects, and you can see their eye movements on top of it. This is a system that, being based on coils and being designed to be uniform and a lot of other constraints, really allows you to record very fine eye movements when the head is free. But it's extremely cumbersome, and it's also invasive. And most of the data we'll show you today come from systems like this. It's called the dual Purkinje image eye tracker. This is the classical, traditional version that you could buy until a few years ago. The subjects needs to be immobilized in order to record their movements here. But then you can go to very fine levels. It's also a system that is quite simple in the way it operates. It's just looking for reflections from an infrared beam. And so it was possible in the last few years to replicate this system in digital form. So nowadays it's just a camera. So it becomes much easier to record the movements. Not only that, in our laboratory, this system is coupled with another device, and again, this is the old version of it, another device that allows us to modify the stimulus in real time according to eye movements. So we gain control of what is the stimulus on the retina of the subject at relatively high rates. This is how the experiments look like. There is this big setup, which is it's big only because we want to position the cameras and position other things next to the subject, but essentially the eye tracker is only the camera here. But the subject is still immobilized in these experiments. And if you do it right, you can record incredibly small eye movements. Look, this is an artificial eye that is rotated with a galvanometer of just 1 minute of arc. You can see that you can record just 1 minute of arc rotations. 1/60th of a degree, 1/60th of your fingernail at arm length. So why all this? Well, by coupling high-resolution recordings of movements with this real-time system for gaze contingent control, we can take control of what is on the retina. Normally, visual scientists, as visual scientists, call the stimulus what is on the monitor. But every subject will look at that stimulus in a different way. So the spatiotemporal flow that they are experiencing is not under our control. With a system like this, you gain control because you can, for example, stabilize it, right? You can move the stimulus together with the eye. A little bit like the suction cap, like the pole that I showed before, but less invasive, I think, for present. And also you can do any kind of motion on top, so you can create a motion on the retina that you want. Now your stimulus becomes this. Okay. So using these kind of devices, we and others have accumulated quite a bit of evidence that the luminous modulations from eye drift matters. The standard idea, the typical experiment comes from something like this: drift modulations enhance fine patterns and the controls that are associated. These are experiments in which we ask subjects to do fine discrimination. Is this noisy grating tilted like this, or is it tilted like that? Simple questions like this. And when you do it normally, right, we put enough level of noise so that it's tricky. You're not 100%. Sometimes you're wrong. But then when we stabilize the stimulus on the retina, what happens is that performance drops down. And they specifically drop down only for the high frequency grating. If I do the same questions at low frequency, where the input changes, the temporal modulations wouldn't be much, right? Drift wouldn't produce much of a change. Then performance remains the same. So this is consistent with the idea that the temporal changes from eye drift contributes. And again, lots of other controls about the directions, et cetera, et cetera. But,
can I ask a quick question?
Yes, please, of course.
So I was wondering, because you said before we have control kind of over our drift, so wouldn't it make sense in such scenario to change our drift speed then to
yeah, it would.
benefit us from that one, but we're not doing that?
No, under retinal stabilization, you usually don't do. I mean, but we throw away a lot of trials. So there is a lot of details in this. So we throw away a lot of trials so that the standard, what we are comparing is similar. So if we see trials in which the subject, we call it, starts chasing the stimulus, right? You start seeing a little bit of larger drift, we typically discard it.
Okay.
So to answer to your question, they change a little bit, but I can't tell you how much they're changing it because maybe our retina stabilization is not perfect, or maybe we put the stimulus a little bit on the side, and so now they start following that versus they're not. But I will show you some other experiments in which they clearly change their behavior according to that. Here is another experiment that perhaps is more controlled, is more recent. Before we had normal viewing and retinal stabilization. Now I'm going to play some intermediate versions of this. So for example, we can reduce the amount of motion just by cutting in half, the motion of the eye, so moving the stimulus with the eye, but at half speed. Or we can move it in the opposite direction, so we can increase the amount of motion on the retina. If I do this, I can control, I throw away that slide, I knew it. Here it is, okay. If you do this, I can control the amount of motion on the retina. I'm calling this the diffusion constant because we always compare Brownian motion, drift to Brownian motion. So you should read this axis as amount of motion on the retina. These different manipulations, they're here, allow you to control the amount of motion that you have on the retina. So you can increase, you can decrease it, you can increase it, you can increase it by a large amount. Okay, and when you do that, you can then measure contrast sensitivity, and here is what we are doing. We are measuring contrast sensitivity for these subjects, and depending on the condition, depending on how much they move their eyes, they have different sensitivity. There is a range in which they peak. Too little motion, too much motion is not beneficial. And interestingly enough, if you just take the stimuli and the traces of eye movements, and you calculate how much of these modulations are available at different temporal frequencies. For example, let's take 5 hertz. It just turns out that the power that is delivered at 5 hertz very closely matches the performance of the subject. There is no modeling involved here. There are no free parameters. There's nothing. I'm just taking the signal from the input on the retina given by the stimulus and the eye movements and looking at what is available around 5 hertz. Okay, and in this case, Howard and Janice are down there. Most of the other people will appear where Martina is in the future. So please keep an eye for those who did the work. Also, 5 hertz is not a particular arbitrary temporal frequency, is not too far off from what P cells would respond to. So if you just look at what is the strength of the signal within the range of sensitivity of P cells, more or less we are there with respect to what we found in the experiment. So spatial sensitivity seems to closely follow the temporal modulations from eye drift. I won't go into this, but you can build models and closely predict contrast sensitivity, not just to space, but also to time if you take into account drift modulation and with minimal assumptions. Then another line of evidence is that the way we change our eye movements, the way we change our drift, is consistent with what we want to do with these luminous changes. And there are several experiments on this. Here is one. When you go to an eye doctor and you do a 20/20 line eye chart, right? You're looking at the high standard for high visual acuity. You pass the 20/20 line eye chart test. Now you can go without glasses. Well, sure, they're measuring how good the refraction is, but indirectly, they're also measuring your own eye movements. Because what we do, you have the feeling that you're reading the optotypes, and you are. What you're doing, you're making very small saccade from one optotype to the next. And by the way, these are incredibly small saccades because they are 10 minutes of arc, smaller than the standard saccade that you would make when fixating at a single dot on average. And on each optotype, then you jitter your eyes, right? You have this drift. But if you look at the characteristics of this jitter, it's different than the characteristics of the drift that you would have, for example, during fixation on a dot. It's different because that temporal filter that I showed you before, the amount of power that it produces, is now shifted, is smaller, it's now shifted toward higher spatial frequencies. So I would say to enhance the signal at the frequency that is relevant, at the frequency of the optotypes. The jitter becomes smaller. As the microsaccades, also the jitter becomes smaller when you're doing an eye chart at the 2020 line. What else? Martina mentioned this. Individual acuity limits are predicted by drift characteristics. We all, no, a few people, not sorry, we all, a few people have 20/20 vision, right? But even those 20/20 visionaries, they're not all equal. Some of them have higher visual acuity, some of them have lower visual acuity. They also all move their eyes very differently. Some of them move them more, some of them move them less. There is an incredibly nice correlation for me between these two things. So you can basically predict what the acuity of each individual is just by simply asking them to fixate on a dot and looking at the amount of their eye jitter. The two things are correlated. And this, by the way, that one extends also to myopes nowadays, we know. Okay, so there is a number of things that suggest that the modulations from eye drift matter. Good, but again, is this all? I would argue not. I would argue that this principle that we just show for drift extends to every type of eye movements. In fact, it extends to every type of behavior. And in fact, it extends to be a general principle of computations beyond what is available on the retina. I won't cover all these aspects that I just mentioned, but I would certainly like to talk about different types of eye movements. Probably the biggest transients come from saccades, right? Apart from blinks, the biggest transients that we are exposed to come from those rapid gaze shifts that we perform so frequently. And I remind you that that these gaze shifts, saccades, are very highly structured, and very specific dynamics. So you can imagine that because of that, they will transform the spatial image into a temporal pattern in a very specific way. And here it is. Okay. So what you're looking at here is the previous slide, the first slide we saw from eye blinks that tells you that all spatial frequencies are transformed into temporal changes in the same way. Then the slide that we saw a little bit later from eye drift that shows that you get this band pass filter. You don't get much temporal changes at low spatial frequency from eye drift. And now I will introduce another curve that is the one from saccades. So what is the input change caused by a saccade? You take the saccade, you reconstruct the movie of the input to the retina from one stimulus to the end to another stimulus. You do the power spectrum analysis, many saccades. That's what you end up with. There is a range. First of all, it's creating more power than eye drift. That's not surprising. You are changing more, so it's a bigger change. And this, by the way, it's a relatively small saccade, a 1 degree saccade. But you're still creating more change than drift, so you create more power. That's fine. But it's also creating less power than a blink. And that also is not too surprising because at very low spatial frequencies, even the largest saccade doesn't produce much of a change. Over a uniform field, I can make as much saccade as I want. I don't produce any change, right? But at low spatial frequencies, an eye blink will always create the same amount of change. Now, there are a couple of things, and Zetuo is hidden there behind. Sorry, Zetuo. There is another couple of interesting things. So first one is that there is still a range in which power grows up as spatial frequency, like we had for drift. Then it saturates, but there is this range of whitening. Something to remember. Now, what happens if I take other saccades? This was a 1 degree saccade. Let's take larger saccade. Well, if I take larger saccades, what I'm doing, I'm keeping the same shape, but I'm pushing it down toward lower and lower spatial frequency. The region where it saturates becomes at lower spatial frequencies. And there are reasons for this in the dynamics of a movement, in the main sequence. You can mathematically show why this is happening. If you take smaller saccades, you bring it closer to drift. So this is a very small saccade here, and now here is drift. And remember, you can also change that curve because I showed you before, you can control drift. So this will be drift during free viewing. This will be drift during the Snellen eye chart test, which is smaller. So basically, you have a continuum in saccade drifts and then beyond are continuing in the curves in which the visual system can be exposed to. Remember, I'm assuming that the visual system doesn't respond to 0 hertz, doesn't respond to things that don't change. It only responds to things that change. And essentially, this picture tells us what is responding to if you assume that there is equal sensitivity throughout those behaviors. Okay? This is the input that is receiving as I'm making saccades or I'm making drift. Does it matter? Well, let's talk about the consequences. And I go back to the small saccade, 1 degree, and I'm going to compare it to eye drifts. Now, the simplest experiment I can do, let's measure contrast sensitivity during fixation. Traditional way, which you can find in many textbooks. Subjects are fixating, they're looking for the faintest grating they can tell. But they're moving their eyes, right, during fixation. If I measure sensitivity at 10 cycles per degree, high spatial frequencies, well, here, saccades or drift produce the same amount of power, same amount of change. I'm not going to expect much change in the trials in which there is only drift or in those occasional trials in which subjects also make saccades. But let's, for example, take a lower spatial frequency. Now here, saccades are producing a little bit more power, stronger input signals. And indeed, if I go back to those trials in which we measure contrast sensitivity, normally what we would do, we would take all the trials and measure the threshold. But if I take those trials apart, those that only had drift and those that also had some small saccades, you see that there is a difference. Now there is more sensitivity when you had a small saccade. Okay, so that's consistent with the idea that saccades perhaps help. Let's elaborate a little bit further. And let's take a slightly larger saccade, 3-4 degree saccade. So now there is a lot more power than drift. Okay, again, I want to measure things at high spatial frequencies, and I'm interested in this case in seeing how vision evolves after a saccade. So I make a saccade, and then I'm going to measure sensitivity immediately after a saccade, or later on during fixation. If I'm measuring sensitivity at high spatial frequencies, a simple model will tell you that what you may expect is an integration of those modulations. You get some transient from the saccade initially, then you get similar transients from drift, so you keep improving. The same model will tell you that if you measure sensitivity at low spatial frequencies, drift here doesn't do anything relative to saccades. And so you would expect that you get initial sensitivity from the saccade and then not much improvements from the drift because it's at low spatial frequencies. And again, this is very similar to what subjects do. It's not only similar, but these improvements is driven by drift transients in large part, because when we stabilize the stimulus on the retina, here you see a drop in performance. Okay? And what about this initial performance here? Well, this initial sensitivity at low spatial frequencies depends in very large part from the saccade transient, because if you do the same experiment, but now you make the saccade land, and then the stimulus starts raising in contrast, and perhaps you make the exposure even much longer now, much longer than 800 milliseconds. Maybe you can do almost 2 seconds of ramping up the stimulus gradually, and then 800 milliseconds exposure. Well, if you do that, now performance at low spatial frequencies drop down. Okay? As if the temporal change from the saccade is important, is essential for seeing low spatial frequencies. But there is more, and please interrupt me at any time. There is a prediction here that visibility will depend on the transient, and therefore will depend on the amplitude of the saccade, because obviously a small saccade will produce a different change than a large saccade. Let's go back to our curve. And, Howard is there behind Martina. Okay. So let's go back to a small saccade, and this will become a 1 degree saccade and a large saccade, a 6 degree saccade. And if you make a large saccade, as before, we produce more power at low spatial frequency. That's the main difference. Okay, so let's think about an experiment like this. in which I'm going to compare the visibility of a probe relative to a reference. Two gratings, I'm going to compare which one is more visible. But here I'm choosing these two gratings wisely. I'm choosing them in a range in which small and large saccades don't make any difference. They produce the same changes. So you can look at the stimulus as something like this. I can make them orthogonal. There is a probe on one axis, and there is the reference on the other axis. The reference is at lower spatial frequency than the probe. And I can design my experiment, I will show you in a second, so that I'm going to test what is more visible. And here, don't expect much of a change. But let's say that I make my probe at lower spatial frequency than the reference. Now, the two saccades should have different actions, and the large saccade should improve visibility of the probe. That's what I would expect. Right. Okay, so how am I going to design this experiment? I take my two gratings, here they are. This one is at 0.1 cycles per degrees at lower frequency than the reference. I'm going to vary systematically the contrast of this, so I create my psychometric function. I can estimate the point of subjective equality. They make 50% of the time they tell me that one is more visible, the other one is more visible. And I can do the same for the other experiments, but now the probe is at higher frequency than the reference. The reference is as before. So the subject will fixate, the stimulus starts appearing very slowly, they don't even realize it. They make a saccade, they can be either a small or a large saccade, and there is a mask. And then they have to tell me, oh, the stimulus was tilted to the left one was more visible than the right one. And so we measure which one, if the probe or the reference was the one. Okay, let's start with this configuration. The reference is a higher frequency than the reference. The two saccades are supposed to give equal power. And in fact, that's the power, that's the strength of the modulations that we get with these subjects. What we are doing here, we are taking the saccades, we are reconstructing the movie, we are calculating how much temporal signals there is in the range of sensitivity of the visual system. Yes?
Sorry, just that I understand this right. Could you maybe go one slide back to the trial design? Like
the experiment.
The experiment, yeah, exactly.
I am trying. No, I'm going forward, sorry.
Yeah, so when they're now deciding left-right, I mean, obviously it's going to be much more faint, but in this case, I guess people see both, right? Like,
yeah, they have to tell me which one is more visible. They see both. They have to tell me, do you see one as more visible than the other one?
I would struggle here, to be honest.
Yeah, it's a difficult task.
I'm not sure what I would say here, left or right.
Yeah. So we give subjects, we prepare them. So we show them many gradation of the stimuli, and we told them this will be the left more visible, when it's very clear, this will be the right more visible, and they learn rather quickly. So, but initially it's confusing. Okay.
Yeah, I think I also would have had a question to this, that I would have expected people to have a bias to either higher or lower spatial frequencies, but when you look at the results, it doesn't look like this, right?
I haven't shown the result yet.
I thought in the...
I will show the power.
Okay.
Yes, each subject are going to have their own biases,
Okay.
but you always compare things. So it's the same subject, same biases, but you're always comparing what happens with a small saccade and what happens with a large saccade.
I see, yeah.
Okay, yeah.
Okay. One question about the pattern. Did I understand correctly that the contrast between the two patterns is randomized? So between trials, the contrast is basically random, but you look over many trials?
So this is done with a method of constant stimuli. So we systematically vary the contrast of the probe to recreate the psychometric function. I didn't put here a psychometric function, but they look quite clean. Okay, and then once we have the psychometric function, we look for the point of subjective. We fit them with a line, with a curve, cumulative Gaussian, and then we look for the point of subjective equality. So the threshold, the 50% threshold. Okay, so this is not the performance, this is just the power. So I make the saccade, I reconstruct the movie, I'm looking at the strength of the input signals, and each of these will be one subject. Essentially what I'm saying is that small and large saccades have similar transits. They're creating similar transits. Here are their performance. Different subjects have different level of contrast sensitivity, but they don't change whether they make a small saccade or the large saccade. Okay, let's however go to the more interesting case in which we have the probe at lower frequency than the grating. Here, I'm expecting a saccade to help because a larger saccade is producing a lot more power than a smaller saccade. Okay, and the performance of the subjects, I wouldn't be telling you this otherwise, is improving with a 6-degree saccade. And this is consistent with all subjects we tested. There is an improvement when you make a larger saccade than a smaller saccade if the probe is at lower frequency than the reference. So this is interesting, but we had this for quite a long time. And I always thought, like, there are so many variables that maybe could have affected this. I mean, can we find a better way to see if this is the temporal change? And Howard had this idea of trying with a slightly smaller saccade. And here is the rationale. A slightly smaller saccade should have the exact opposite effect of what I showed you before. Let's work this out. So before we saw a 1-degree saccade and a 6-degree saccade. In the low probe condition, the large saccade helps. And that's what we saw before. The large saccade helps. Let's make just a slightly smaller saccade. We only go from 1 degree to half a degree, 0.4 degrees. But what happens here? The 0.4 degrees is pushing this range of sensitivity of the reference in the, what we call the whitening zone, in the range in which this grows, still is not saturated yet. So both power for the low probe and the reference have decreased, but they have decreased by the same amount. So I would expect that in this case, performance don't change relative to a 1-degree saccade. And indeed, that's what happens. Performance don't change. Even though I'm producing with the transients less power for the probe and less power from the reference, but I'm producing less power for both of them in the same way. Let's look at what happens with the high frequency, for the high probe, higher frequency than the reference. Now, all saccades produce the same transients for the probe. But for the reference, whereas the 1 and 6-degree saccade had produced the same power, now the smaller saccade produced less power. So you should unmask the probe. You should make the probe more visible. And here is the performance. In this case, before, with the 6 and 1-degree saccade, we found no difference because both of them, we think, were in the saturation region. But now when we look at the smaller saccade, performance goes up. And this, again, is consistent with what we would expect from temporal changes. I'm probably dragging this too long. Let me just mention that I can take other transients, other type of behaviors, and still see similar effects. For example, and this is going to be very compact, pursuit. I'm tracking something as it's moving on my screen, and so that on the retina, it remains sort of stationary, right? But it's not really stationary because pursuit is not perfect. In fact, if you look at the amount of motion of the stimulus on the retina during pursuit, is not so different from the amount of motion that you have during fixation. During fixation, you're taking an intersaccadic fixation, you have small saccades and then the jitter of the eye. So the input, the retinal motion, will have small fluctuations, then a saccade, small fluctuations. During pursuit, you have something quite similar. Now you're tracking the object, right? You're pursuing the target, but you have some catch-up saccades, they're called, but they're not all catch-up. Sometimes you look backward, okay? It's the normal way of tracking the stimulus, so that you have small saccades and then temporal changes because the stimulus is not perfectly stationary on the retina. Now, in these experiments, same rationale. We're going to look for the temporal power that is produced by the motion and how performance is in discriminating objects. I will just summarizing without telling you what the experiment actually is, but they have to do fine discriminations. The more power, the better they are in doing, in resolving the stimulus. Let's go back to blinks, which is what we started with. Blinks, we said, generate equal transients at all spatial frequencies. But how can I test it, whether that matters? Well, I'm going to compare it with what happens when the eyes remain open. But when the eyes remain open, the eyes are jittering. And that jitter enhances high spatial frequencies. So blinks enhance everything, but the jitter of the eye enhance high spatial frequency. I'm expecting that blinks would improve performance at lower spatial frequencies if I compare trials with blinks and trials without blinks. And so we took the frequency range, which is in that range, and performance, discrimination performance, when we blink, in the trials in which we blink, are typically better than in the trials when we not blinks. And this happens irrespective of where the blink is coming from. You can do the blink yourself, or you can be exposed to a stimulus change that is kind of similar to what the blink would do, so a change in contrast of the stimulus. Okay, I think I can just wrap up. So what did I say? I said a few things. First, I proposed my idea for how space is encoded in the visual system. I call it active space-time encoding. It's a strategy for representing space. It's based on motor behavior that shapes the input signals, distributing spatial information into the temporal domain. And because of the way I move, there is a range. This signal, use of this signal is efficient. I didn't talk too much about efficiency, but you've seen this whitening, and we can talk more during the questions. And there is a growing body of evidence supporting the idea that the visual system uses these signals. So now I would like to leave you with some food for thoughts and implications. But if this is true, well, here we have another function of eye movements, right? Eye movements are not just generated to fovate, but they're generated also to produce transients. I know Martin has talked about that as well. They had a nice review. There are some species out there that don't have a fovea, they still make eye movements. Maybe they're doing it for this reason. This makes, from an engineering point of view, it's a strategy that makes sense, not just because of the whitening, etc., which we didn't discuss in detail, but also because it's flexible. I have a somewhat fixed machinery that is there operating the input, but now I can feed that machinery with different signals by moving in different ways. So I can easily adapt representations to the task. I can tune representations to the task by changing my behavior. If you open textbooks of vision, that's something that always bugged me. There are chapters on spatial vision and chapters on motion perception, and then you read the physiology and you see all neurons respond to motion. So how is that coming together? Well, I think this is sort of unifying framework for putting motion perception and spatial vision, because visual perception always relies on change. I didn't talk at all about how the decoding, how the interpretation of these signals work. But I want to mention that there is evidence that we have quite a lot of knowledge about how the eye is moving, extraretinal knowledge of how the eye is moving at the level of eye drifts. And this is one paper that convinced me of that. There is now an analogy with other sensory modalities. Like other modalities, vision needs to be studied in the context of motor behavior. And I presented quite a lot of data here in this review, and not all the people that I highlighted are here, but I presented data from Bing Yang, from Zetuo, from Howard. Is Janice here? Janice, here she is. And I presented at the beginning that data from Rui Tao. I hope I'm not forgetting anybody. Here, Rui is the guy that really made our eye tracker possible. And I think I covered everybody. And a lot of these ideas are in collaborations with Jonathan Victor. So thank you.